Thanks for the introduction, and thanks for having me here. It's my first time at this conference, and it's yeah, really fantastic meeting in a fantastic city. So what I'm going to tell you about is actually a story that is quite deeply sort of like linked into the actual biology. I think I realized today that I don't think I actually have like any methods in this talk, in these slides. There's methods sort of like underneath. But actually when it comes to the sort of spectrum of topics at this conference, my background is certainly on the biological end of things. But hopefully I'll be able to inspire you a little bit thinking about some of the kind of questions and problems that we think about linking genetic effects on gene expression dosage into cellular and also physiological phenotypes. So the general space where my research program and a huge part of human genetics research operates in is in the dilemma of functional interpretation of genetic variation. So as you all know, during the past 15 years, there's been this enormous success story in human genetics being able to link genetic variation to complex diseases and traits, and also actually rare diseases as well. But the problem that we have now, and that is emerging as perhaps the most pressing problem actually in using these very, very rich data, is the gap in understanding what are the molecular and cellular mechanisms that actually mediate these associations between teeny tiny genetic perturbations, which is naturally occurring variation in human populations, and very complex physiological phenotypes. And my lab in particular has been interested in sort of mapping how genetic variants link to gene expression and other transcriptome traits to bridge this gap. In addition to these kinds of little bit, I don't know, let's say medically I guess medically focused aims of trying to understand mechanisms of disease associations, there is also potential applications in molecular precision medicine, so using these kinds of functional readouts to support your genetic diagnosis or a prediction. That's not something that I'm going to talk about today, even though we've worked a little bit in that space as well. But what is really the focus of my of this talk today is using these data sets and many different approaches actually to understand the functional genetic architecture of human traits. Like what are actually some of the general patterns of how all of these things come together, what types of genetic variants via what kind of mechanisms link to human traits. And one thing that I want to highlight is that this kind of this field, thinking about functional interpretation of genetic variation, is very much actually at the intersection of two classically quite distinct areas of biology. There is the sort of the population genetics and the GWAS folks, etc., who really think about genetic variation, and there is a very, very rich sort of family of different statistical methods in that realm. And then there is the sort of molecular genomics folks, people and projects like ENCODE that are thinking about how does the blueprint of the genome sequence produce a functional cell and a functional organism, and how does that work, and where are the enhancers, and how do TS bind, and da-da-da. And we're actually here in the middle taking kind of like both experimental and statistical methods from both directions. But so here we kind of like try to partition this sort of variant to function dilemma a little bit further. The starting point is of course kind of asking what are the genetic variants associated to diseases and traits. GWAS, rare variant studies are kind of like they're doing that. I'm not going to talk about that today, and I generally don't actually work in that space too much. I just use those results. And the second question is, what are the effects of those variants on their target genes in terms of do they affect its expression, splicing, somehow kind of like what happens there at the cis-regulatory molecular space? And then there's the even bigger question downstream of like, so if we understand how these variants affect in particular gene dosage, gene expression levels, how does that actually bring us to physiological phenotypes or cellular phenotypes for that matter? What is the sort of relationship between functional gene dosage and that gene's downstream function? And there is a lot of pretty basic sort of data that indicates that this kind of a relationship is probably quite complex. And this is something that I'm here illustrating just by thinking about the classical models of dominance. So recessive dominant genes and variants, this is like basically probably the first genetic phenomena that we teach to school children, where like if you're heterozygous for a recessive gene, you lose half of that gene's functional dosage and you're fine. If this happens in a dominant gene or a dominant variant, you lose half of the gene dosage and you're screwed. And then there is things kind of like in between where you may have something in the middle. But actually, we don't, even though this is, I mean, as a phenomenon, has been known since Mendel, we don't actually know how this happens. I just made up these curves. In addition to kind of like not understanding why certain genes are recessive or dominant, we also don't know actually, like let's say you have a dominant gene and you lose half the gene dosage and terrible things happen, horrible disease. What happens if you lose 10% or 20%, or what if you gain 10% of that gene dosage? What is actually the shape of these curves? We really don't know, and we also don't know kind of why. We do have a lot of evidence also from this, just sort of what we know about the general architecture of rare and complex disease from the past, I don't know, 10, 15, 20 years of genetic research using modern tools, that gene dosage is really a key driver in both of these occasions. So we, of course, know that common traits and diseases are typically, most of the variants that we find in GWAS studies are fairly common in the population, each of them with a teeny tiny effect size. And then monogenic Mendelian diseases are very low frequency variants with high effect sizes per variant. And there is also kind of like a spectrum of sort of like what are the functional effects or where in the genome these variants are located, with rare Mendelian disease variants being typically coding loss of function variants, GWAS variants being about 90% of them in the non-coding space in the genome, likely affecting gene regulation. One thing that I want to note here is that I don't want to kind of like completely ignore the fact that there are variants that affect protein structure, and that is also a very important topic, but I'm not going to talk about that today, and we don't work on that so much. I'm here kind of like really focusing on the sort of gene expression dosage space. But so when we think about these kinds of coding variants in rare disease and regulatory variants in complex disease and kind of how does that actually link to gene dosage, that link is actually quite complex. So there is often people kind of tend to have this thinking that like, okay, if you're coding variants, they affect maybe gene structure or something, and non-coding variants affect gene regulation, but actually both variants via different types of mechanisms that are partially captured by more sophisticated annotation of these variants can actually contribute to changes both in protein structure and then in gene dosage. And the sad thing is that making this kind of inference, taking a variant either in the coding or non-coding space and saying that, okay, this is this variant's effect on gene dosage, and we can actually infer this, predict this, is not something where we actually are as a field. And that's why we need to measure these effects. And that is why this is such a big field, because the question is actually quite difficult. There are three sort of fundamental approaches that the field is currently taking to tackle this variant to function challenge. The first one here is perhaps the one that is more familiar to the audience here, basically using the population genetic association approach that projects like GTEx have used. So basically you collect both genome sequencing or genetic data from a population sample and RNA sequencing data or another molecular phenotype, and then you associate the two, look for genetic associations for molecular phenotypes. There is also the slightly more modern, newer approach that became kind of like scalable maybe about 10 years ago, where we can do very scalable in vitro perturbations in a cellular model. So you, for example, grow a cancer cell line on a dish, and you use tools like CRISPR or MPRA or others to perturb genome or its function, and then you see what happens, and you basically associate those two. Then there's also the kind of like, let's say, I call it the encode way here, where you just kind of, you do functional characterization of a lot of different cell types. You don't really kind of like perturb the genome necessarily here, but you kind of like integrate this data and potentially can use this for a prediction of molecular effects. We don't really work in that space, so I'm not going to talk about that. But instead, I will talk about these two approaches here and how we can actually bring them together in interesting ways. To start from the population genetics approach, I've been fortunate to have the opportunity to work in many different large projects that have done integration of very, very large genome and transcriptome data sets, ranging back, I don't know, I guess over 10 years now, analyzing 1,000 genome cell lines and different types of things in little bit different contexts. The largest study in this kind of a space, looking for genetic associations for gene expression and other molecular traits, is the Genotype Tissue Expression Project, or GTEx, that I'm sure many of you have heard about. It is now, it went on for about 10 years. We published the final set of papers a few years ago, and now there is the next sort of phase of GTEx, developmental GTEx, that is ongoing or getting started now. I'm not going to talk too much about GTEx, but just to kind of like very briefly sort of say what are the sort of like insights that are or data sets that are coming out of this project. In the final stage of GTEx, we had over 17,000 tissue samples from over 50 tissues, from over 800 donors, with RNA sequencing from all the tissue samples, and then genome sequencing from each of the donors from blood. And this allows you to then map eQTLs, expression quantitative trait loci, and splicing QTLs, where you basically look for genetic associations between genotype and these molecular traits. And one of the sort of main things that kind of comes out if you start looking at GTEx data or any other larger eQTL data sets is that basically all human genes have common genetic regulatory variation in the population. This type of variation is extremely common. Splicing variation is slightly less common, but still extremely widespread as well. And in the next few slides, I'll kind of like basically glance through a bunch of sort of little bit general insights that link these types of data into various assessments of the role of this kind of regulatory variation and in complex disease, and then also rare disease, and bring these things together. I want to highlight that I'm kind of sort of summarizing here probably like, I don't know, 10 years of work by a huge community of people, just to kind of like bring you into some of the questions and gaps that we are still left with even after analyzing large data sets like GTEx. So when it comes to using these kinds of eQTL and splicing QTL data sets in analysis of complex disease, there is these are from the final GTEx paper, but there are many equivalent studies in the community showing that overall there is a genome-wide enrichment of GWAS signal. You can measure that as heritability enrichment or in many various different ways. There is a robust enrichment of this signal. There is a lot of debate in the community if that enrichment that we see is actually a lot or a little or as much as we expected, or it's a little bit like glass half empty or half full type of a debate that is ongoing. But that's this is still kind of a very robust pattern that we see. In addition to that, two other kind of classical approaches to link eQTL data or molecular QTL data and GWAS studies is co-localization and TWAS, or transcriptome-wide association studies, with co-localization basically looking at, do these two association loci, one for a molecular phenotype and one for a physiological phenotype, do they share the same causal variant? And TWAS then using sort of genetic predictors of gene expression to associate that with GWAS phenotypes. Those things provide putative sort of hypotheses for causal genes, causal regulatory mechanisms for thousands of individual loci. So that's kind of sort of baseline of how one looks at gene regulatory variance in the context of complex disease. In the rare variant space, we have a slightly different situation because actually in silico inference of variants that have much stronger impact on gene function, that are typically rare in the population, that contribute to rare disease, these in silico inferences are actually pretty good. We can actually just use the genetic code to identify that this is a premature stop variant, or this is a variant in a splice site, etc. But these predictions are not perfect, and also the inference of what is actually the molecular consequence of those variants is not always perfect, and this partially contributes to the gap that we have in genetic diagnosis as well. Transcriptome data sets have been used to bridge that gap, so basically looking at the impact of rare, putatively sort of gene perturbing, gene disrupting variants in what is the impact of those variants at the transcriptome level. Using GTEx data sets, we've been part of doing this type of work starting from way back when. It wasn't, it was 2015, that 9 is a typo. But anyways, looking at how premature stop variant trigger nonsense mediate decay that basically just degrades those mutant copies of the transcripts. And then more recently, in the final stages of the GTEx project, using multiple different transcriptome metrics to look at how rare variants actually perturb the transcriptome. So this helps us to kind of understand what happens there, how rare variants contribute to transcriptome variation. But still, we are kind of in this sort of situation where I mean, in human genetics, there are two quite distinct communities. There is the sort of rare variant community that mostly looks at coding variants with large effects in the context of Mendelian disease or cancer, often using exome sequencing studies. And then there's the complex disease community that thinks about GWASs, complex trace, association mapping, often using SNP array data sets. And what I kind of want to argue here in the next couple of slides and then moving onwards is that we actually want to think about the joint effects that these different types of variants have on gene dosage, and use that as sort of like a point of convergence that allows us to think about disease risk in a way that doesn't lead to this kind of somewhat arbitrary partitioning to rare and common diseases and variants. And in the next few slides, I want to discuss what are some of the systematic efforts to study common and rare variation under this kind of a joint framework. So first of all, in a study that we published a few years ago, we were able to basically use EQTL and allelic expression data in GTEx, capturing mostly common variation in gene expression, to basically calculate for each gene what is the variance in gene expression due to genetic variation in cis. And this is a metric that varies between genes, with patterns such as haploinsufficient genes or genes with key biological roles being generally sort of constrained, like having relatively little genetically driven variation in their expression, and some genes, non-essential genes, other genes being highly variable. And in this work, we actually use this kind of variance that we derive from common variation to prioritize rare, putatively disease-causing variants by kind of asking when an individual having a rare variant is sort of outside of the normal spectrum of gene expression variants. So that's one way how we can integrate the two, and we're actually continuing to do quite a lot of work in this space, also in collaboration with Peshman Mohammadi, who used to be a postdoc with me and is now at University of Washington. Another way to think about how these different variants kind of come together is to actually ask how do they kind of converge. So if we imagine a situation now that we have very rich data sets of both GWAS data looking at common variant associations to diseases and phenotypes, and also rare variants with increasing amounts of whole genome sequencing data, we can ask that, like, okay, so if you have sort of like one variant that is associated to a complex trait, and then there are other variants that are also sort of like potentially affecting the same gene as this variant, do they actually do they have similar effects on these complex traits, suggesting that they might be kind of like mediated by the effects on the same gene? And this is something that we analyzed in GTEx. To make a very long story and a lot of analysis, to kind of like cut that very short, we basically see that when we look at situations where one gene is affected by multiple eQTLs, or is affected by an eQTL and a splicing QTL, or it's affected by eQTLs, and then there are also rare variants in that gene, we see these kinds of like sort of these effects on GWAS lining up, indicating that these kinds of genes with sort of multiple lines of independent evidence are, of course, kind of gold standard causal genes for these traits. These kinds of genes also allow us to think a little bit more sort of explicitly about kind of like what are truly the joint impacts of rare and common variants in the same gene. In one sort of line of work that we've been pursuing in my lab with some published work and some things in bioRxiv, we have been analyzing how basically joint haplotypic effects of rare and common variants end up having sort of joint effect on functional gene dosage, and how that associates to sort of purifying selection and also human phenotypes. But probably the more relevant way of thinking about it, especially considering the sort of my dosage to function curves in the beginning of this talk, is the concept of allelic series. This is something that Robert Plench and some others more in the sort of pharma space have been talking about a lot. That is really kind of like along that same gene function or gene dosage to phenotype type of a curve is what this concept or these approaches are looking at. And the idea is basically that when you have different types of variants that perturb the gene's dosage or the gene's function at different levels, if you are able to line those things up, actually measure how much they affect that gene's function, and then you can also see how each of them affect that phenotype, this can actually be quite insightful for many different purposes. The only problem is that there are very few empirical examples of people being actually able to put this kind of an allelic series together. And the reason for that is that functional genetic variation is actually quite sparse in the human population, especially for genes that we're most interested in because of purifying selection. So when we think about GTEx eQTLs, so we have these for like every gene, and for many genes we have multiple eQTLs. That's nice and great, but most of them have teeny tiny effect sizes. So here the x-axis is the absolute allelic fold change, a metric that we developed with Peshman several years ago now. But basically, like it's just like expression fold change just for genetic variants. And here is on log 2 scale, so 1 would basically mean 2-fold change in gene expression. And you can see kind of like here after 0, the curves just sort of like shoot up. Depends a little bit on the sample size, what's the specific sort of like how the curve goes, but in any case, most eQTLs have teeny tiny effect sizes. And then, I mean, we do have those rare variants that have very strong impact on gene expression dosage, things like they disrupt splicing or introduce stop codons, etc. But I mean, by definition, rare variants are rare. And we're typically with this kind of a situation that we might have some common eQTLs with teeny tiny effects, and then we have some rare variants in very few individuals with strong effects, and it doesn't really allow us to sort of map out this sort of like a dosage to function curve for the vast majority of human genes. We still want to try, and I'll maybe in a few years I'll be able to tell you more, but it's not easy. And this kind of a caveat is something that we've actually discussed a little bit more explicitly in a review that we wrote with Dan MacArthur a few years ago. Basically, how genetic analysis in human populations is obviously limited to studying the existing genetic variation in the population. There are also some other confounders of kind of complicated, various type of sort of mess that you get when you deal with real populations. However, while cellular models in vitro using those kinds of experimental perturbations have their own weaknesses in terms of like you're studying some cancer cell line growing on a dish, but one of the biggest advantages, at least in my opinion, is that you can actually perturb any loci with different variants and different effect sizes pretty much as you want. You're not constrained to studying the spectrum of variation that we have in a natural population passing through natural selection. So this brings me basically back to this kind of functional gene dosage curve and thinking about how can we actually map this if we turn to these types of experimental perturbation approaches. And this is a project that we've been pursuing now for a couple of years in my lab in New York, led by Julia Domingo, an absolute superstar of a postdoc. And we're now sort of like writing up this work. It will be coming to a preprint archive near you sometime later this spring. But currently still unpublished work for the remainder of the talk. So what we basically wanted to do in this study was to fine-tune transcription factor dosage with CRISPR to discover these kinds of non-linear regulatory responses and basically map out for these transcription factors how does their dosage link to downstream phenotypes. And here we chose, so this is just a pilot study that we started doing. We'll be expanding on this, but the results are, I think, already quite interesting. So here we chose 4 different genes, 3 of those genes being transcription factors, TET2 is not. And these 4 genes, we basically want to modulate their expression levels with CRISPR. And then what we use as a sort of like a phenotype readout in this study was gene expression of basically genes that are downstream in the regulatory networks from these 2 genes. And so we selected a bunch of genes for that, looking at those like downstream transcriptome effects based on another study that we did together with Neville Sanchana's lab, led by John Morris, where we actually looked at GFI1B and NFE2 in a sort of GWAS enhancery type of an approach. That's a different story. But so basically we wanted to here analyze, we could have looked at the entire transcriptome at the single cell level, but we actually chose to look at targeted gene expression of a large number of genes, but not all genes, to get a really, really high precision, good quality readout of the expression of those downstream genes. So the experimental setup is this. So basically we are using the CRISPR-I, CRISPR-A, so CRISPR activation and inactivation machinery in the K562 cell line, coupled with CRISPR guide RNAs to increase and decrease gene expression. So for each of the 4 assist genes, we use different types of guide RNAs in these 2 cell lines, kind of like in a pooled experiment, to modulate their dosage up and down. So basically, when we have guide RNAs that target the transcription start site, this is kind of like more like the classic perturb-seq type of a design where you can, at least potentially likely, should be introducing a pretty strong effect inactivating or activating the gene. There is also a different sort of like a design where you have guide RNAs at the transcription start site, but they have mismatches in them, so that they're basically their efficacy is reduced. And this is a previously published approach for titrating gene dosage, especially downregulation of gene dosage gradually. We also tested something that, at least to my knowledge, hasn't been tested before, basically tiling guide RNAs, so moving away from the transcription start site, ±1 megabase. And then for 3 of these genes, we also knew some enhancers for these genes that are more distal, and we targeted those. So that's basically what was the kind of the perturbation of these genes. And then the readout is done with excite-seq, which where we use hashtag olico, so basically a readout of surface proteins to distinguish whether we are looking at a CRISPR-I or CRISPR-A cell in a given cell. We have a readout of what is the guide RNA. So here we have each guide RNA, one guide RNA per cell. And then we have a targeted hybridization-based approach to have very high precision readout of expression levels of 92 targeted genes. So that is the 4 genes that we're looking at are cis genes and then 88 other genes. And we have several hundred cells per guide in each of the 2 cell lines. So what does the data look like? Looks like this. So here I basically have the expression change of each of the 4 genes on the y-axis for the different types of guide RNAs that we have. The NTC is non-targeting guides, so these are our negative controls. And what you can see here is changes in gene expression in various different ways to different extents for the different genes and also for the different guide RNAs. With the tiling approach, so basically guide RNAs moving away from the transcription start site, giving us the widest range and also really kind of nice ranges here going up-regulating gene expression in a gradual way. So this is the approach that we think is the best for titration of gene dosage with CRISPR. And altogether, when we kind of like sum up all these data, this is the kind of like range of gene dosage that we have for each of the 4 genes, with GFI1b having the widest range of very strong downregulation of gene expression and also substantial upregulation. We've also done some analysis to show that this is not due to just sort of like cells having different frequencies of binary up sort of knockdown effects. That checks out. But to sort of to dig a little bit deeper, so what kinds of guide RNAs actually affect its dosage effects? This is a little bit more sort of if you want to study these kinds of or do these kinds of studies, we can see that there are effects from these distal enhancers. But zooming into this kind of space where we to do this tiling, we see expected kind of effects where generally the effect kind of like goes smaller as you move away from transcription start site, and also the epigenetic marks for CRISPR-I and CRISPR-A are slightly different in terms of where do you find the stronger effects. But the thing that we were most interested in this study was thinking about, so how does this gene expression variation link to gene expression response in trans? So looking at the expression of these 88 other genes in the genome and thinking about this as a downstream phenotype. And here, so there are previous approaches that have done a little bit similar things. So the sort of perturb-seq type of analysis, crop-seq is another sort of protocol for a similar thing, where you basically look at transcriptional response with single-cell sequencing as a function of gene knockdown or knockout. So you have a very drastic silencing of these genes, and then you see what happens to the transcriptome. Versus our approach is that we have these 4 genes and multiple different variations in that gene's dosage, and then we see what happens. So I want to just like explain this plot because you'll be seeing a few of these. So what the data looks like is that, so for each of the 4 genes, we basically look at its dosage on the x-axis, and then each of these 88 genes on the y-axis. And here each dot is given a couple of hundred cells with a given guide RNA and basically the median up or down regulation of each of the 2 genes there. And in addition to having just the dots there, you can see that there is a line there, and that comes from a sigmoid model that we fit into these data. So to basically, model these patterns, and it generally provides a pretty nice fit and allows us to also derive multiple different parameters of what is actually going on here. So what is the slope of the change? How sort of steep that curve is? What is the lower and upper asymptote of these sigmoid curves? So basically, how big is the range that we see here? And then when does it cross 0? And we can see that there is, for many of these guide RNAs, there is generally a much better, for many of these genes, there is really a much better fit with a sigmoidal fit compared to a linear fit. So we really see plenty of nonlinear dosage responses here, as we would expect. I mean, sigmoidal fit is kind of like the natural sort of like a dosage response that you would expect to see in a biological system, so we were really glad to see that. We do see diverse types of curves here, so there's obviously genes that don't like kind of like move anywhere. There is different types of patterns of more or less steep up and down regulation. The nice thing here is that, like, even though you can see the kind of the sigmoidal fit here, it's not that you really need to kind of sort of squint your eyes and think that like maybe there is a curve there. The data is actually quite clean and quite nice. This is partially thanks to the fact that we did targeted single-cell RNA sequencing that gives us really precise measurement of gene expression levels. I don't know if any of you noticed, but there is, on this slide, there is one gene here that is actually not a monotonic response, so there is a curve here, and then it goes down both in the kind of left and right end of this. So there are a few exceptions to this monotonic dosage response, and those we model just with a simple lowest fit. We will need to have more data to really understand why these things happen, but they are there too as an exception. But so altogether across the entire data set, this is kind of like what the data looks like. This is only for GFI1b, where we have kind of here in the raw data, this would be the kind of like the dosage changes of GFI1b with our guide RNAs targeting that gene's transcription site and around. And this is kind of the resulting transgene response. Now having that sigmoid fit for all these genes allows us to basically smoothen out these patterns, not just for GFI1b, but also for MyoB and NFE2. TET2 is a gene that has so little transcriptional response that we basically don't analyze it very much. But one can see that there is sort of like different patterns here where you have some classes of genes where, like, let's say GFI1b and MyoB have kind of the downstream response is the opposite. There are also situations where they line up. Some genes respond more or less, etc. We can summarize the transcriptional response across these 88 genes as basically just kind of like a mean absolute fold change across all of those genes, as this kind of like a composite transcriptional perturbation phenotype. And we see some interesting patterns here as well, especially for GFI1b, where we have sort of like clear non-linear parts here, and then it shoots up, and it's interesting to think about what's going on there. We can further think about different types of sort of endophenotypes that we can derive from these gene expression profiles. This is a little bit sort of, let's say, exploratory analysis, where we basically kind of look at genes that are known to either increase or decrease in expression in certain cell lineages, and then we look at the change in the expression of these gene sets and summarize that as kind of like a marker of potential differentiation potential that is kind of like that we see in the transcriptional networks going into different cell types. And here, I mean, this is kind of, again, exploratory preliminary data, but what we are thinking of is basically linking this kind of dosage of transcription factors to cellular phenotypes with an interest, again, in these kinds of like nonlinear responses. And we have ongoing experiments to actually measure differentiation instead of just trying to infer this kind of differentiation potential from expression data. But so looking at all these like pretty rich data of how the dosage of 3 different transcription factors affects the expression of genes downstream, I just want to ask, so are there some properties of these transgenes that might correlate with, for example, their role in disease? So this is an example. Oops, something has happened here. I've lost the dots. So now you see just the fits. This is interesting. Yeah, well, whatever. Yeah, so these are basically the kinds of patterns that we see just for an example set of 8 OMIM genes, where you can see that as the dosage of these transcription factors change, they respond in various different ways. And some of these OMIM disease genes some of them are recessive, some are dominant, and one can sort of like start to think about what might happen downstream. We do see that there is both for OMIM genes and for GWAS genes, there is some enrichment of nonlinear dosage responses, potentially suggesting that genes that are important for downstream phenotypes and disease are basically enriched for this kind of finely tuned transcriptome response. But we wanted to take this analysis a little bit further and ask that it's like, okay, so are these kinds of responses linked to some other properties of these genes, such as conservation, constraint, enhancer redundancy, their role in the regulatory networks, what's the binding of these TFs around these genes, etc. Kind of basically asking that it's like, okay, so let's say these genes that don't respond at all, are these like super constrained, conserved genes that are very tightly regulated? So we put together this kind of like a ginormous matrix of like just collecting various different types of properties of these genes and kind of analyzing this. And when we looked at the correlation of those different factors, so they are now here on the y-axis with the different properties that we derive from our sigmoid fit, and kind of like asked that, so are these correlated? Or is it, for example, so that genes that are annotated as potentially haploinsufficient have a smaller range of gene expression change? There are some patterns, and we're like, okay, so maybe there's something here. And then we looked at the other 3 transcription factors and really see that there is no clear and consistent link. Nothing here really kind of lines up in a way that is consistent. So that was a little bit disappointing in the beginning. Oh, my emojis have broken, oh no. But basically, so after that early disappointment, we started scratching our head a little and basically realized that what this means is that these kinds of, the way genes respond to different regulatory stimuli, different sort of triggers in these cells and different sort of cellular states is highly specific. And that these kinds of properties that genes have in terms of, let's say, their enhancer regulation and their constraint is not easily linked to how they respond to different cellular stimuli. And what this means basically is that we do have a lot of work to do to basically understand why, let's say, dosage-sensitive genes, how do they actually regulate their gene dosage in a way that doesn't lead to terrible consequences to the organism. So to summarize, we basically have here a new sort of CRISPR system, a little toolkit to gradually modify gene expression. And this is a tool that we're now using in a couple of different projects. The kind of little patterns that we have learned thus far help us to design these experiments better. And we generally get a pretty good resolution, although the range of modification that we can introduce varies a little bit between genes in ways that we don't understand yet and need to collect more data to analyze. We see these widespread nonlinear and nicely sigmoid dosage responses in trans that make a lot of sense and provide rich data for analysis of how gene dosage links to its downstream functions. And we see that disease genes are potentially enriched for nonlinear responses. And altogether, like now we have been basically looking at how gene dosage links to just kind of expression of downstream genes. We have ongoing experiments to analyze how it links to cellular phenotypes. And then there is, of course, the sort of like the whole phenotype space from just downstream gene expression all the way to different organisms. Of course, I mean, with humans, you're not going to do these kinds of dosage titration experiments to look at human phenotypes. And this is why I'm not abandoning human genetics here. With human genetics, we can potentially, with approaches like Allelic series, actually link some of these mechanisms to human phenotypes as well. And why we would want to do that is actually multiple different reasons. So if you think about sort of precision type of an approach where we know from complex and rare disease studies that gene dosage is one of those key drivers underlying human phenotypes, you would potentially want to kind of like actually put your individuals on this spectrum when you want to think about genetic diagnosis and prediction of that individual's course of disease. In drug development, there is a lot of interest in this type of work. That's why they have been talking about Allelic series for 10 to 15 years, because there is obviously this kind of like an interest in thinking about what is this sort of like a modification that you would want to have in your drug target so that you actually have the drug efficacy left, but potentially some side effects kind of reduced, and thinking about sort of multiple phenotypes at the same time is also something that we're interested in. And then finally, if we think about just basic molecular biology, a lot of molecular biology, probably most of it, is based on gene knockdowns and knockouts, where you really hit those genes, hit those biological mechanisms with a sledgehammer. And then on the other side, in let's say in human genetics and in epidemiology, we're mostly talking about very tiny perturbations of the system with genetic or environmental sort of modifiers and variation. And I think that we really need to start to think about understanding that link and understanding when do those sledgehammer type of things actually give us interpretable and useful biological insights, and when do we need to actually tune these systems and perturb them only very so slightly. I think that this is generally a very, very exciting time for integration of observational and experimental functional genetics, where we can do this molecular analysis and genetic analysis of large cohorts, where we can capture cells and tissues in their native state, where we have potentially a more direct link to clinical applications. But we do suffer from some of the complexities of observational data, where you don't have sort of the clarity of experimental perturbations that you can repeat and replicate. And then there is also some of the complications of, let's say, getting resolution of cell types and different molecular modalities. And then in large-scale perturbation experiments, some things are very clear and nice. At the same time, we're talking about cellular models on a dish. So even though there is a lot of power and a lot of hype in this kind of like, let's just kind of CRISPR everything type of an approach, I do think that we need to maintain the connection to analysis of actual humans and their genes and their tissues to really capture insights that allow us to understand, not just kind of make predictions in the precision medicine space, but also understand molecular mechanisms. And I'll finish here with acknowledgements, because thanks go to Julia, who has been leading the CRISPR work, and then a lot of people in my both labs, a lot of lab alumni, collaborators, especially collaborators in GTEx, and our funders. And I will also mention that we're looking for postdocs, actually, both in Sweden and in New York. I'll finish here and take some questions. Thanks.